Hello and welcome to today's lesson looking at the structure of human blood. In the previous lesson we looked at transport in animals and we spoke about the need for mass transport systems. We spoke about closed circulatory systems and open circulatory systems, and we compared how circulatory systems work in fish, insects and mammals. In today's lesson, we're going to look at the structure of human blood, so specifically looking at mammals, specifically humans. By the end of this lesson, you should be able to name the parts of the blood and what each part does. You should be able to explain what hydrostatic and oncotic pressure are, and you should be able to evaluate the differences between the compositions of the blood, tissue fluid, and lymph. So first things first is you need to understand, and I'm going to put this in red because oftentimes it's forgotten, that the blood is a tissue. It's made up of lots of cells and lots of substances. So the blood is a tissue made of many cells and substances. And substances, we'll just neaten that up a bit. Substances, okay? Now the blood consists of multiple parts, and if we spin blood through a centrifuge, we would get a separation that looks something like this, okay? So we can say that the blood consists of dot dot. We've got what are known as erythrocytes, E-R-Y, T-H-R-O, C-Y-T-E-S, or more commonly known as red blood cells. Okay, erythrocytes is the scientific name for red blood cells. Now, erythrocytes, or red blood cells, carry oxygen. It's as simple as that. All they do is carry oxygen. The second part of the blood that we need to talk about is the plasma. Now, plasma is the liquid that makes up the blood. Okay, so the liquid part of blood is known as plasma. So the liquid part of blood that also carries carbon dioxide and urea. So that carries carbon dioxide and urea. Urea is a chemical that it contains all the waste substances from cells. Now, as you can see in this diagram here, it says that the plasma makes up 55% of your blood. So the majority of your blood is plasma, while 41% is your red blood cells. So plasma makes up 55% of the blood. So 55% of the blood. Okay. The next part are what are known as platelets. P-L-A-T-E-L-E-T-S, platelets. And platelets are large fragments of cells that helps the blood clot. Okay, so large fragments of dead cells that helps the blood clot. That helps blood clot. Now, what we mean by blood clotting is when blood dries at wound sites. So if you happen to cut yourself, you would have clotting at the wound site to form a scab and stop any extra pathogens entering your body and to prevent blood from escaping. So platelets play an important role in the immune system. They help as a physical barrier against external pathogens. And finally, white blood cells. White blood cells are cells that bring about immunity. So cells that bring about immunity. So your blood is a tissue. It's made up of the following parts: erythrocytes, which are red blood cells. They make up about 41% of your blood. Plasma, which is the liquid part of your blood that carries carbon dioxide and urea. It makes up the majority of your blood at 55%. Platelets, which are large fragments of dead cells that helps the blood to clot. And white blood cells that bring about immunity. Together, platelets and white blood cells make up about 4% of your blood. Okay? Now, the blood is responsible for transporting substances such as molecules, nutrients and hormones. It's important for the repair of damaged areas and for the maintenance of your body temperature and pH. So I'm going to put here, in fact I'm going to put it in this colour, the blood is responsible for the transport of substances These include things like molecules, they include nutrients, and they can include hormones. It's also responsible for the repair of damaged sites. And it's helpful in the maintenance of body temperature and pH, and plays a role in the maintenance can't spell maintenance, that's right, isn't it? Plays a role in the maintenance of body temperature and pH. So your blood is responsible for the transport of substances, such as molecules, nutrients, and hormones, the repair of damaged sites, such as where you cut your skin or anything like that, and it plays a role in the maintenance of body temperature and pH. Now, substances that are dissolved in the plasma of the blood can leave capillaries and move into surrounding cells. These substances usually give the blood a high solute concentration, meaning that water moves into the blood. Okay? So we'll get that written down in red because it's important. We can say that substances dissolved in the plasma can leave capillaries and move into surrounding cells. So if we've got a capillary, substances can leave it and move into surrounding cells. So if I've got a blood capillary down here, so this is my capillary, okay, that's my capillary. Inside that capillary, we've got some solutes dissolved in the plasma. So solutes dissolved in plasma. I know that doesn't say that, but it's the best I can do. So these are your solutes dissolved in plasma. These can leave the capillary and move into surrounding cells. So these solutes that are found inside the capillary can diffuse from inside the capillary into surrounding cells. Okay? So these are cells. And we've said what we've said here, basically, that substances can leave the blood plasma in the capillary and move into surrounding cells. Now, wherever these solutes are found can change the osmotic potential or the water potential of the blood. So if there are lots of solutes in the blood, water is more likely to move into the blood down an osmotic gradient. Whereas if solutes move out of this blood, water is likely to leave the blood and go into surrounding cells. So we can say, and continue in red, that when solutes move into the blood, this lowers water potential causing water to move into the blood with it. Causing water to move into the blood with it. So when solutes are in the blood, it lowers the water potential of the blood and water can move into the capillary. When solutes move into surrounding cells, the opposite happens. So when solutes move into surrounding cells, this raises the water potential of the blood of the blood causing water to leave the capillary. Causing water to leave the capillary. Now remember, everything in my lessons that are important is written in red. So this is a really important concept. We said that substances dissolved in the plasma can leave capillaries and move into surrounding cells. So once blood is in the capillary, substances in the plasma can move into surrounding cells, but at the same time, substances from the cells can also move into the blood. Okay, it can happen both ways. When solutes move into the blood, this lowers water potential, causing water to move into the blood with it. So when you get lots of solutes moving into the blood, this makes the blood more concentrated, and therefore water moves down its osmotic gradient into the blood from the surrounding cells. At the same time, if solutes move out of the blood into surrounding cells, the opposite happens. Water will follow the solutes where they move because you're lowering the water potential of the cells surrounding it. Okay? Right then. So I'm going to refresh the screen. Obviously you can pause this video or contact me should you be confused about something. We now need to talk about two keywords. The first keyword is the term oncotic pressure. Oncotic pressure. O-N-C-O-T-I-C pressure. Oncotic pressure is when water moves into the blood via osmosis. So this is the pressure created when water moves into the blood. So pressure created when water moves into the blood. So this is usually due to excess solutes in the blood, which lower its water potential. So we can say that this is due to increased solutes in the blood causing water to move into the blood. Causing water to move into the blood. The other type of pressure that we're going to talk about today is hydrostatic pressure. Hydrostatic pressure. Now, hydrostatic pressure is the pressure generated in blood from heartbeats. When your heart contracts, it increases the pressure inside of the blood vessels. This is known as hydrostatic pressure. So we can define this as the pressure generated or created in the blood, in blood from heartbeats. So when your heart beats, the pressure inside of the blood vessel increases, and we call this hydrostatic pressure. Okay? Now, if we think of an artery and a capillary and a vein, we have your heart, which, and I'm going to do this as a single loop to make it a bit easier. We do know that it is a double closed system, it's not a single closed system. If this is your heart, blood is pumped out of your heart through arteries. Okay? So arteries take blood away from the heart. Arteries then break down into small vessels known as capillaries. And capillaries form what are known as capillary beds. They split up into millions and millions of blood vessels that all intertwine from the artery. So this is a capillary bed. Okay? Capillary beds then feed into veins which take blood back to the heart. So we've got an artery, a capillary, and then a vein. Blood moves in this direction around the circulatory system. So blood will be moving from the through the arteries to the capillaries, through the capillaries to the veins, and then the veins take blood back to the heart. Okay? Blood, when it leaves the arteries, is under high pressure. So we can say that blood leaves the arteries or the heart through arteries under high pressure. Okay, so blood leaves the heart through the arteries under high pressure. Once blood reaches the capillaries, it loses this pressure. So as it moves through the capillaries, the pressure is lost. So pressure is lost from the blood at capillary beds. Once the blood gets to the veins, the vein has to have lots of adaptations that bring the blood back to the heart under low pressure. So we can say the blood in veins is under low pressure. So we've got this blood that is being pumped from the heart through arteries under high pressure. This would be a lot of hydrostatic pressure because the heart has contracted and pushed the blood through the arteries. Once it reaches the capillaries, the blood loses its pressure. Okay? So if we were thinking of this as a sort of linear scale, and I can't remember what colour I used. Was it this one? That's right. When we've got arteries feeding into capillaries, which then feed into veins, like so. So we've got arteries feeding into capillaries, and then we've got capillaries feeding into veins. Where the artery meets the capillary, there would be high hydrostatic pressure, much higher than oncotic pressure. Okay? So we can say that hydrostatic pressure is higher than oncotic pressure. Okay, so the hydrostatic pressure is higher than oncotic pressure. For this reason, because the hydrostatic pressure is higher than oncotic pressure, water is pushed out of the capillaries. So we can say water, or fluid, in fact, we'll change that to, apologies. Fluid is pushed out of the capillary. So this high hydrostatic pressure pushes water out of the capillary. Now, as the blood moves through the capillary bed, hydrostatic pressure is lost. Therefore, where the capillary meets the veins, the opposite happens. So oncotic pressure is higher than hydrostatic pressure, and for this reason, fluid is pushed back in to the capillaries. So fluid is pushed back into the capillaries. Okay? So oncotic pressure is the pressure created when water moves into the blood. This is due to an increased amount of solutes in the blood. Hydrostatic pressure is the pressure created in the blood from a heartbeat. Every time the heart beats, blood is pushed out of the heart through the arteries under high hydrostatic pressure. The blood then reaches the capillary beds, where hydrostatic pressure, which would be on our diagram here, and on this one would be here. Hydrostatic pressure is higher than oncotic pressure, so fluid is pushed out of the capillaries. When we get to the capillary end, the vein end of the capillary, that's where we get this bit. Okay? Oncotic pressure is higher than hydrostatic pressure, and fluid is pushed back into the capillaries. Now, a much neater way of showing this looks like this, and it's a diagram I stole off Google earlier. So when we're talking about a capillary, the hydrostatic pressure is higher at the arterial end. So we can say that arterial end of capillary bed dot dot, we can say that hydrostatic pressure is high. We can say that oncotic pressure is low. Oncotic pressure is low, and therefore water or fluids are pushed out of the capillary. So fluids are pushed out of the capillary. At the venous end of the capillary, so venous end of the capillary or capillary bed, we can say that hydrostatic pressure is low, oncotic pressure is high, oncotic pressure is high, and therefore fluids are pushed into the capillary. So fluids are pushed into the capillary. I'm going to do that in red because my OCD won't stand for that. Fluids are pushed in or into the capillary. So at the arterial end of the capillary bed, hydrostatic pressure is high, oncotic pressure is low, and therefore fluids are pushed out of the capillary. At the venous end of the capillary bed, hydrostatic pressure is low, oncotic pressure is high, and the fluids are pushed into the capillary. Now the fluids that are pushed out of the capillary form what's known as tissue fluids. So it's a key term, tissue fluid. So it's T-I-S-S-F-L-U-I-D, tissue fluid. Now tissue fluid is the solution surrounding the cells of a multicellular organism. Okay, so the solution surrounding the cells of a multicellular animal. Okay, so it is the solution surrounding the cells of a multicellular animal. Now this is produced at the arterial end of a capillary bed when the fluids are pushed out. So we can say, in fact I'm going to put it in red, that this is made at the arterial end of a capillary bed when fluid is pushed out of the capillary. It's pushed out of the capillary. Okay? So when the fluid is pushed out of the capillary at the arterial end, remember, because this hydrostatic pressure is high and oncotic pressure is low, this forms what's known as tissue fluid. Now, this is basically the same as plasma, except it doesn't contain any erythrocytes or it doesn't contain any proteins. So it's similar to plasma, without the erythrocytes or proteins. Okay, so it doesn't have the erythrocytes and it doesn't have the proteins. Now, a lot of this fluid returns back to the capillary. So at the venous end of the capillary, when oncotic pressure is high, most of this fluid returns back to the capillary. So 10% of this of tissue fluid re-enters the blood at the venous end of the capillary bed. The other, sorry, that shouldn't say 10%, that should say 90%. 90% of it returns to the capillary bed, okay? So 90% of the tissue fluid that enters the blood is pushed back into the capillaries. So 90% of it goes back into the capillary. Tissue fluids are pushed out, 90% of it returns, and then the question is, what happens to the other 10%? So we can add in red at the end here, because it's important. The other 10% becomes lymph, L-Y-P-H. Now lymph, L-Y-M-P-H, lymph is defined as modified tissue fluid that is collected in the lymph system. So modified tissue fluid that is collected in the lymph system. Okay? Now essentially, this is similar to plasma, but it doesn't have oxygen or nutrients. So it's similar to plasma, without oxygen or nutrients. It's transported in lymph vessels, which have lymph nodes in them, which add lymphocytes to it. So it has lymphocytes added to it from the lymph nodes. So lymphocytes are added to it by lymph nodes which transport the lymph via lymph vessels back into the blood. Which transport the lymph via lymph vessels back into the blood. Right then. So at the arterial end of the capillary bed, the hydrostatic pressure is high, the oncotic pressure is low, and therefore fluids are pushed out of the capillary. At the venous end, the oncotic pressure is higher than the hydrostatic, therefore fluids are pushed back into the blood. When the fluids are pushed out of the capillary, they become what's part of known as the tissue fluid, which is a solution surrounding the cells of a multicellular animal. It's very similar to plasma, but it doesn't have erythrocytes and proteins. 90% of this tissue fluid returns back into the blood. The other 10% is taken up by the lymphatic system. It's taken up by lymph vessels and lymph nodes, where lymphocytes are added to the tissue fluid or to turn it into lymph, and the lymph then returns back into your blood, okay, near your aorta. Lovely. So I'm going to refresh the screen now. It seems a shame because it's like a work of art, isn't it? I think we can agree. And we'll move on to the last bit of today's lesson. So, erythrocytes play a major role in the blood. A major role of the blood is to carry oxygen. So, a major role of the blood is to carry oxygen. Okay? Now, erythrocytes have a lifespan of about 120 days. They're constantly being replaced by your bone marrow. Essentially, your liver breaks down old and dead erythrocytes and it is added to your poo. That's why your poo is brown in colour, because it's made up of all these dead, broken down erythrocytes. Okay? So we can say that erythrocytes have a lifespan of about 120 days and are replaced by the bone marrow. By bone marrow. Okay? Now, red blood cells contain a molecule known as hemoglobin that carries oxygen, and it's what makes erythrocytes red. It contains iron that allows it to bind to oxygen. So we can say that erythrocytes contain hemoglobin, Haemoglobin, hemoglobin that carries oxygen and makes the blood red. Okay, carries oxygen and it makes the blood red. Now, when hemoglobin binds to oxygen, it becomes what's known as oxyhemoglobin. So we can show this in a word equation. We can have hemoglobin, or HB. So that's my hemoglobin. Can be added to oxygen. So added to oxygen, O2, or oxygen, in a reversible reaction to form a molecule known as oxyhemoglobin. So oxyhemoglobin. Okay, and oxyhemoglobin has the symbol HB brackets O2 4. So oxyhemoglobin contains 4 oxygen molecules. So each hemoglobin can carry 4 oxygen molecules. So oxyhemoglobin can carry 4 oxygen molecules. Now, the reason it can carry 4 is because hemoglobin contains 4 peptide chains, each with iron. So hemoglobin contains 4 polypeptides. How you spell polypeptide, is it? 4 polypeptide chains, each containing iron. Okay, so each of the polypeptide chains contain iron. Now, when one oxygen joins to the hemoglobin, the molecule changes shape. Now, what this means is that it allows the next oxygen to join. So the joining of one oxygen to hemoglobin causes it to change shape, which allows the other 3 to join. So when the first oxygen joins to the hemoglobin, comma, it changes shape. Full stop. This change in shape increases its affinity for oxygen and allows the other 3 oxygens to join the hemoglobin. So this change in shape increases hemoglobin's, HAEMOGLOBIN, hemoglobin's affinity for oxygen allowing 3 more to join. So once the first oxygen joins to the hemoglobin, it allows more to join to it. Once blood gets to the tissues, the process is reversed, and when one oxygen leaves the hemoglobin, this causes the other 3 to leave. So we can say that this is reversed at the tissues. Now, what we mean by affinity is how attracted a molecule is to another. So affinity is how attracted one molecule is to another. Okay? Now, you'll hear a lot about what are known as oxygen dissociation curves. Okay? And this is essentially a posh way of showing what we've just spoken about. So, what this is saying is, depending on the amount of oxygen in a given area, will show how much oxygen is taken up by the hemoglobin. Okay? So, when we're at the alveoli in the lungs, the first oxygen molecule binds to the hemoglobin and the hemoglobin changes shape. This makes it easier for other ones to join. Okay? At the tissues, the opposite happens. An oxygen leaves the hemoglobin to go into the surrounding tissues that need it for respiration, causing others to leave the hemoglobin. Okay? So at high partial pressures of oxygen, hemoglobin has a higher affinity for oxygen, and therefore oxygen moves into the red blood cells. So when we're at the lungs, there is a high partial pressure of oxygen, there is lots of oxygen, therefore the oxygen will move into the blood and bind to erythrocytes. When we're at the tissues of the body, the tissues of the body are using all of the oxygen up, so they don't have much oxygen available, and therefore the oxygen leaves the blood and moves into those cells. So if we're looking at a disassociation curve in the lungs, there is lots of oxygen, so there is lots of oxygen, and therefore oxygen, or O2, is taken up. In fact, I'm going to get rid of that first. So there's lots of O2 shown by the partial pressure here at 80. Okay? We can say that here oxygen is taken up by erythrocytes. erythrocytes, erythrocytes. In the exercising tissues, there isn't much oxygen. That's not going to show up, is it? I'll do this colour. So there's not much oxygen. Okay? So little oxygen. This oxygen partial pressure is just saying how much oxygen is available in those tissues. Okay? Therefore, oxygen is released by erythrocytes and given to the surrounding tissues. So O2 is released by erythrocytes and given to cells. Here, on the right-hand side, affinity is high. So, on this side of the graph, affinity for oxygen is high. Whereas on this side of the graph, affinity for oxygen is low. So on this side, we've got affinity for oxygen is high, and that's in the erythrocytes. And on this side, affinity for oxygen is low in the erythrocytes. So on this side of the graph, this is where the erythrocytes are taking up oxygen from the lungs, and on this side is where they are dropping them off to the exercising tissues or any other tissues. So if you think about it, as you go up, you get more and more oxygens added to this hemoglobin. Okay? They've used double the numbers to represent individual oxygen molecules. Okay, bear in mind oxygen is diatomic and it exists as two molecules joined together. So here you've got 0 oxygen bound to the blood. Here you would have 2 oxygen molecule, well, 1 oxygen molecule joined to the blood. Here you would have 2. Here you would have 3, and here you would have 4. As oxygen partial pressure increases, the uptake of oxygen by erythrocytes is higher. So, hopefully by now you can tell me the parts of the blood and what they do. You can explain what hydrostatic and oncotic pressure are, and you can evaluate the differences between the blood, tissue fluid, and lymph. Should you have any questions about today's lesson, please get in contact with me via the usual means and I will get back to you as soon as I can. If not, I will see you in the next video. Take care.